Today I will let you know intermediate filament, that is another type of cytoskeleton. So if we talk of the general properties of the intermediate filament, its diameter there is a range 9 to 12 nanometer, that is in between the microfilament as well as the microtubule diameter. And if we talk of in which type of the tissues intermediate filament are more in number, so let me tell you, in those tissue that have a function of protection, means I could say protective function है ना, tissues that shows of the protective function, or other ways we can say that most of the time epithelial cell है ना, say that they just protect the distortion of the epithelial cell. Let me explain you with an example. Whenever you just do stretching or yoga, at that times these intermediate filaments are there that just protect your epithelial cell from any kind of distortion, is it? So इंटरमीडिएट फिलामेंट्स की बात करें तो मैक्सिमम टेंसाइल स्ट्रेंथ है, और अदर वर्ड्स में अगर हम बोलें तो ये ऑफकोर्स टेंसाइल स्ट्रेंथ की हम बात कर रहे हैं तो हम ये बोल सकते हैं ये स्टेबल ज़्यादा है, स्टेबिलिटी इनमें ज़्यादा है. अल्टरनेटिवली हम लोगों ने जो कल डिस्कस किया था, स्टेबल ज़्यादा है तो लेस डायनामिक है एज कम्पेयर टू बोथ ऑफ देम, ऑल्दो हैं डायनामिक बट लेस डायनामिक है. यहाँ पे देर आर नो कॉन्स्टिट्यूएंट सब यूनिट दैट बाइंड अ न्यूक्लियोटाइड. बेसिकली this can be explained like that there are no ATPase and GTPase, there are no motor protein, and a very important thing to remember, that is they are not found in all eukaryotes है ना, they are more prominent in the animal cell, totally absent in the plant and fungi है ना. So this is a point to remember. Then we can see here the assembly of microfilament, how they just assemble. So primary building block is dimer. What basically happen, you can just see here the dimer and they will combine together, end to end combination or the association is there, and they will lead to the formation of the tetramer है ना, this protofilament. So interlocking will be there, you can just see है ना. Likewise two more will be here, and together these they form a tetramer. So these four protofilament give rise to one protofibril, further four protofibril. It means in one protofibril how many protofilaments are there? 4 है ना. So total 16 protofilaments are there. 4 protofibril lie side by side, and they just give rise to the intermediate filament, that is of 10 nanometer. So next is classes of intermediate filament है ना. Different classes are there, they are classified on the basis of the distribution. So the very first is nuclear, then vimentin like, and the epithelial. So in nucleus we just observe a nuclear lamina. So there are that lamin protein, that is a type of intermediate filament only. There are different type of, although lamin A, lamin B and lamin C protein. Then second is the vimentin like. So it could further be subdivided in vimentin, desmin and GFAP है ना, where they are just observed, according to that they are just named. So vimentin type leukocyte blood vessel in the endothelial cell as well as some epithelial cell, mesenchymal cell है ना, these they are common in such tissues. Further in skeletal and cardiac and these we observe desmin. Cardiac as well as the smooth muscle cell we observe desmin, and they just are responsible for giving strength and organizing them. Here further is glial cell है ना. And so next is the epithelial. So here I will let you know there are different type. We are going to just see type 1, type 2, type 3, type 4. So type 1 and type 2 is epithelial type only, है ना. Type 3 is vimentin type. I will let you know, there is a table in this lecture only. So epithelial, you can just see type 1 keratin acidic type and type 2 keratin basic type. These are more common in the hairs and nails, है ना. Keratin is a well known example of the intermediate filament present in hair as well as the nails. Yes, these are largest as well as insoluble in water and the organic solvent. So this is the point to remember. Full form of GFAP is glial fibrillation acidic production, है ना. Acidic protein. These are common in the glial cell. So here is an example of the skeletal muscle contraction where the although one more is there, axonal neurofilament. This is also is a type of this intermediate filament only. So neuro, it means this will lie in the neuron, है ना. In the neuron only we are going to just observe such neurofilament. Although there are different type neurofilament, M is there, H is there, L is there, है ना. So there are different types present in the neural cell. So here we are going to just see the skeletal muscle contraction. So they are stimulated by nerve impulse. Nerve impulse, what basically they just trigger an action potential in muscle cell plasma membrane. How action potential is triggered, that is another thing, है ना. Here we just are looking skeletal muscle as an example of intermediate filament. So I'm not going in detail how this basically the membrane potential change and the action potential is generated. So basically once the action potential is generated in membrane, they just travel down and it leads to the invagination of the plasma membrane known as the transverse tubules. And further these tubule like structure you can just see here in the figure, है ना. Once the action potential generated, what will happen, it will just travel and it will just stimulate the opening of the voltage gated calcium channel, you can just see. And there will be increase in the calcium cytosolic in cytosol calcium concentration will be increased. Then further once the concentration is increased, and from where these calcium will be released, from the sarcoplasmic reticulum, that is a specific type of the endoplasmic reticulum of the smooth muscle cell. So once these calcium concentration is increased, what basically will happen, this membrane is known as presynaptic membrane, है ना. So this will fuse with these are the presynaptic membrane and they will just fuse with it. And then further what will happen, once the fusion take place, acetylcholine will be released in these space, that is known as synaptic space. And here are the receptors that will receive these chemicals, that is the acetylcholine. And once they receive, action potential will be generated along the muscle fiber, you can just see here, है ना. These are protofilaments, है ना, all 13 protofilaments are there. Once action potential are generated, this will travel into T tubule, and then further you can just see calcium release from sarcoplasmic reticulum and it will bind to troponin. Two things are there, troponin and tropomodulin, है ना. So this you can just see how basically this calcium release will lead to the binding to troponin. And once they just bind to troponin, everything is also mentioned here also, है ना. This elevated calcium, they will induce change in the troponin as well as the tropomyosin, है ना. Troponin type you can just see troponin I, C, T, and this tropomyosin is basically a rope like and it will bind 7-actin, and this tropomyosin will bind with troponin only for functioning, you can just see here. These are thick filament, that is the myosin, and thin filament is the these actin, है ना. So you can just see the distance between two is the sarcomere. So like this only the contraction takes place in the skeletal muscles. So what basically happened, some points are mentioned here in different colors, so these are important. So what if suppose there are no calcium, so the interaction will be blocked. How it will be blocked? The stropomyosin, what they will do, they will block the myosin interaction with the actin, and once the myosin interaction with the actin will be blocked, there will be no contraction observed. And if the binding of calcium is there with the troponin C, it will further triggers the movement of tropomyosin to new site on filament, and as it will trigger to move further, it will expose the myosin binding site on actin, and hence the contraction will take place. So this figure you can just see different types of the that I just have discussed only, that epithelial and vimentin type and all. So here you can just see type 1, as I told, type 1 is the epithelial, and there are two type, acidic as well as the basic, है ना, type 1 and type 2. Further third one was the vimentin type, in which type of the tissue they are just common in the fibroblast, astrocyte, in endothelial cell, in muscle cell, है ना, these are common. You can just see the name of the intermediate filament proteins that are involved in the different tissues. Further type 4 type in the neuron, that is the axonal, I just have discussed, neurofilament L, M, H, all these are there. Then further the lamin type, and in the nucleus, although they are present in all cell type, है ना. Then further type 6 is the in the lenses, है ना, filancin, facinin, CP49. So these are the different types of the intermediate filament, or I could say the different proteins that are involved in the formation of intermediate filament. They are categorized according to their position, है ना, where they are located. So that's all. Thank you for watching. I hope this video helped you.